Okay, hi everyone. It's Dr. Flanagan. We're going to go ahead and move on to the next section, the last section, 13.12 through 13.18 of Chapter 13 from your Campbell Biology textbook. As a reminder, everything comes from Pearson Education. So we talked before about Hardy-Weinberg equilibrium, and if the 5 conditions for Hardy-Weinberg equilibrium are not met in a population, then allele frequencies will probably change. But the thing about it is that mutations are rare and random. Mutations are rare and random, and they oftentimes have little effect on the gene pool. Non-random mating may also change genotype frequencies, but usually it doesn't have any effect on allele frequencies either. So remember when I talked about a string of DNA, a big long string of DNA inside the cells. There you might have these little sections that code for proteins like this. Okay, let's say on this one string we have 3 sections that code for proteins. Protein number 1, protein number 2, and protein number 3. And a mutation happens. And let's say a mutation happens right here. That mutation does nothing. Okay, and then let's say another mutation happens here. Does nothing. Another one here. Does nothing. Something has to fall just randomly within one of these sections in order to have one of these small little areas, in order to have it have an effect on the population or have an effect on the individual. Then it has to be a favorable gene in order to really have an effect on the population. So it has to be something in a rare occasion where that mutation happens to be better for the population. And maybe this is the reason why we have all this kind of garbage DNA, all this extra stuff, is just to dilute out the effect that mutations have on genes. So we know mutations happen. How can we avoid it happening in genes every time? Well, we put this extra garbage here that allows mutations to happen without really affecting the population. What that kind of boils down to is that there's mutations and non-random mating. They may have a little bit of an effect, but the 3 main causes of evolutionary change are natural selection, genetic drift, and gene flow. Alright, now, I don't expect you to know what either of these 2 mean yet, but I'm going to explain it. And this one we've talked about in some detail, so you should know that one pretty well. But we're going to go, and I'm going to make some more points about natural selection before we get into these other 2. So natural selection, this is actually one of your fill in the blanks here. Natural selection, if individuals differ in their survival and reproductive success, natural selection will alter their allele frequency. Okay, so if individuals differ in reproductive success, natural selection will alter allele frequencies. And if you guys remember, an allele is an alternate form of a gene. And frequencies are just the amount of them in the population. So let's just go number, maybe we call it percentage in the population. And so if you have one form of a gene that is very successful, then we expect that one to increase in frequency in the population. Or if you have a gene, alternatively, if you have a gene that's maybe not so good, but works, and is kept around because it works good sometimes, but maybe not right now, then that will decrease in frequency in the population. So there you go. That's natural selection in a nutshell. So if you think about the iguana population that we talked about before, the individuals with webbed feet, that was genotype w, they might survive better and reproduce more offspring because they are better swimmers and better at catching food than individuals that lack having webbed feet. And so that's just one example from the book of when a frequency might pick up. If having webbed feet helps you swim better, and being able to swim better gives you better access to mates or better access to food, then that frequency of that being in the population will increase. All right, next process that leads to evolutionary change is genetic drift. Okay, again, another one of your underlines. This is number 2. Now, in this process called genetic drift, that's the next underline, chance events cause allele frequencies to fluctuate unpredictably from one generation to the next. If we go back to our sequence here that I showed you before, where we have these small sections where we have genes, and if we get a mutation that happens right here in the gene, and that gene's still functional, then it sticks around in the population. And then you might get 6 or 8 different versions of the gene in the population, kind of in the gene pool of the population. And then as individuals breed, there's just the potential for some of those to become more concentrated, even if it doesn't provide us a selective advantage, okay, natural selection advantage. So sometimes those those versions of a gene. Should they all be equal? Sometimes some of them will randomly become the more common in the population, and almost always drift will happen. It just kind of, even in big populations, drift can happen, but the smaller the population, the more impact genetic drift is likely to have. So in a very small population, if we only have, let's say there's only, in the whole population, let's say there's only 5 different versions of this gene, and there's only 2 versions of this gene, and there's only maybe 6 versions of this gene, then the likelihood of one of these 5 to become fixed in the population is a little higher. Likelihood of one of these 2 to become fixed in the population, a little higher. Okay, as long as the population is smaller. Certain things like big catastrophes, hurricanes, floods, fires, something like that, they may kill a big number of the individuals in the population, leaving a very small group that survives. And in that small group, then the genetic makeup is likely not to be exactly the same, or it's not likely to be a very representative of the large group that was lost. And so, in those cases, that's an example of drift being forced to happen. Okay, an example that we call the bottleneck effect. Okay, so the bottleneck effect leads to loss of genetic diversity when a population is greatly reduced. I mean, it's kind of like shaking marbles through the neck of a bottle. Here in this population, we have a large number of green, large number of purple, but we also have some orange marbles. And then if you lose a bunch of individuals, those left inside the bottle, then only maybe some, like a lot of purple and only one green, survive to the future populations. Okay, and then that's not representative of what the original population was, and we call that change between those 2 populations the bottleneck effect. After the population is reduced like that, genetic drift then may continue for generations until the population is large enough for fluctuations due to chance to have less of an impact. So for a very long time, it may be that certain alleles get fixed in the population until the population is big enough and mutations have accumulated for long enough that change can occur. The reason that we talk about the bottleneck effect and we need to understand the bottleneck effect is because our activities like hunting or habitat destruction, like cutting down big hunks of the Amazon rainforest and things like that, that could create very severe bottlenecks for other species. Something that's going on right now is with the polar bear. And a lot of people are talking about this, is as there are less and less, or as there are smaller and smaller ice caps and there's less ice breaking off for them to hunt from, the polar bears aren't doing so well. And so the populations are getting reduced, and because the populations are getting reduced, all that's left are very specific genotypes. So there's not as diverse, or not as a genetically diverse population as there once was. Okay, next up, number 3 is gene flow. Allele frequencies in a population can also change as a result of gene flow, okay, where a population may gain or lose alleles when fertile individuals move into or out of a population, or when gametes, such as plant pollen, are transferred between populations. So this is all about migration into or out of a population. If you lose a bunch of individuals, that's kind of the same thing as a bottleneck effect. If a lot of individuals leave all at the same time, or if a bunch of individuals come in, that's going to add a lot of new alleles that could really alter the population. Gene flow tends to reduce differences between populations, so it tends to kind of homogenize large areas. So to counteract the loss of genetic diversity remaining in this specific species of prairie chickens, the scientists, they added a big swath of birds, 271 birds from other states, into the Illinois population, which successfully introduced new alleles, increased genetic diversity, and made a better, more healthy group of prairie chickens.
These prairie chickens are strutting their stuff, hoping to catch the eye of the gals. Fancy footwork. And a bit of clucking. But the ladies have seen it all before. They like their chicken tough. The gents have got to puff themselves up and duke it out. feet.
Now, the issue with that is that sometimes individuals or individual populations are adapted to certain environmental types. So the ones in Illinois might be different than the neighboring states around Illinois. And then that would lead to maybe other issues like a less genetically healthy population than what was formerly there. But the strategy in this case worked really well. New alleles entered the population, and the egg hatching rate improved dramatically. So that's one way that the researchers have helped bolster populations. Genetic drift, gene flow, and mutations could each result in microevolution, but only by chance could these events improve the population's fit to its environment. In natural selection, only the genetic variation produced by mutation in sexual reproduction results from random events. The rest of it is not random. Okay, so the process of natural selection in which individuals are more likely than others to survive is not a random process. It is a process where the environment parses out and selects certain individuals to do a better job than the others. So because of that kind of sorting, natural selection is the only one that consistently leads to adaptation. So only natural selection leads to adaptation. Now, I say consistently because other types of change, evolutionary change, gene flow or genetic drift or any of those could potentially lead to an adaptation where an individual is better fit to survive in the environment or populations are better fit to survive in the environment. But natural selection consistently always leads to an adaptation of some sort. So there you go. But the thing about natural selection is it always is a reduction of genetic diversity. Mutations add new alleles, gene flow adds new alleles. Natural selection is a situation where certain groups of alleles are selected for in the environment and other groups of alleles are selected against. And so other groups are not as successful as the selected individuals, and so only a small portion of the population survives better. So it's always a reduction in genetic variation. What's really cool is that adaptations have some really kind of amazing examples. So let's just look at this one bird here. This is the blue-footed booby. Okay, blue-footed boobies live in the Galapagos Islands. They're super big. They're like 3 feet tall. They have these big webbed flippers, and I'm going to use a different color because their flippers are the same color as the pin that I always use. So they have these bright blue feet. Those are so that they can swim through the water really fast. What they do, and you can see this streamlined body shape, there's this really smooth shape to its body with this big long tail. What they do is they get and they fly really, really high, like 75 feet above the water, and then they dive into really shallow water as fast as they can, and they hit the water, and then they use that big tail as a brake. They open that big tail up, and they can stop like just several feet above the surface, and they can grab fish that are usually really fast that other birds couldn't get. So there's an example of a lot of adaptation that's occurred in one type. A little bit more in natural selection. The common phrase for natural selection is struggle for existence. You'll see that a lot, or survival of the fittest. But both of those are misleading because typically you take those to mean that there's competition between individuals. However, reproductive success is very subtle and passive. It's individuals just tend to do a little better because of this or that, or the other. It doesn't mean that other individuals aren't reproducing at all. It just means that some individuals just happen to be doing a little better. And the ones that do just a little better produce more offspring, and then those offspring, or that allele frequency is favored, or those alleles are favored. What you have to think about instead of this struggle for existence, this is maybe not the best way to think about it. Maybe the better way to think about it is to think about relative fitness. Okay? And relative fitness is the contribution of an individual to the gene pool of the next generation relative to the contributions of others. So all the individuals in the population are breeding, but relative fitness tells us that some individuals breed maybe just a little bit better, producing just a little bit more offspring. They go on to the next generation, and that is what makes them more successful in future generations, or more successful to future generations. Natural selection can affect the distribution of phenotypes in a population. So there are 3 different types of natural selection that we're going to talk about just briefly. First up is stabilizing selection. In stabilizing selection is where the intermediate phenotype is the favored phenotype. So the one's kind of in the middle. And so it's stabilized if you think about a normal curve of a phenotype. Let's say it's like height, okay, in a population. There are not a whole lot of short people. There's a whole lot of people in the middle of the spectrum, and then there's not a whole lot of tall people. Okay, so height's a good example. And stabilizing selection would be like where the people right in the middle of the population, okay, right in the middle of that normal curve, they do the best, okay, and so there's more of a contribution of those individuals to future populations than the ones on either extreme, okay, so it's stabilized towards the center. And then there's also directional selection. This is where one of the extreme phenotypes is favored. So let's say, in this same example, let's say the individuals on this end of the spectrum, the smaller, shorter side of the spectrum, they're the ones who are favored. Then you'll have less of these tall individuals that move on to the next generation, more of the ones on one side, and like that. So you have this directional shift of the mean of the population from kind of a medium height to kind of a shorter height. So that's directional selection. And then the last one, and actually let me change the pin color here. And the last one I'll use pink is disruptive selection. And disruptive selection, is where the intermediate phenotype is not good, okay, and so that pushes kind of bidirectionally towards the phenotypic extremes. So you'll have very tall and very short individuals, and so that's kind of. And I know that this plot, this thing over here, is kind of turned into a bit of a mess, and so let's go ahead and just move on to something a little bit more clean so that you can see it in a little bit easier. Stabilizing selection, the ones in the middle are favored. The ones in the extremes out here don't do as well. Okay, so those are not good. In directional selection, then you have a mean shift, okay, of the population from one direction to the other. And then in disruptive selection, you have from a unimodal population to a bimodal population, where the mean hasn't shifted, but the intermediate phenotypes don't do as well. Okay, so those are the three types of selection. Then we have another type of selection, sexual selection, which may also lead to phenotypic differences, but in this case, it's typically between males and females. So if you guys have ever seen like ducks or birds in general, the males are always really pretty, and the females are always kind of drab in color because females are very choosy, and the male has to be a little prettier so that the females will choose them. So sexual selection is a form of natural selection in which individuals with certain characteristics are more likely than other individuals to obtain mates. So in many animal species, males and females have secondary sexual characteristics, and those are noticeable but not directly associated with survival or necessarily reproduction, but those characteristics are very obvious, like the tail feathers of a peacock, or the masseter muscles of a gorilla, or just in general the size of a male gorilla versus the female gorilla, muscle mass and things like that. So all of that leads to, or all those differences between the two populations, or between the two groups of males and females, that's called sexual dimorphism. Sexual dimorphism, and probably the best example of that, and I don't have a picture of it right now, but there is a little ant, and it's actually quite big in the South, called a velvet ant. Okay, and the Southerners call the velvet ant a cow killer because they're huge and they hurt really bad. It's one of the most painful stings by an insect in America. And the females are these big, really kind of cool looking things, and I'll show you guys a picture here in a second. But the males are just these tiny little, they look like little Drosophila, little flies, little fruit flies, and they just buzz around and they pick up the female and they fly her away and they mate with her, and that's the way it goes. And that's one example, really striking example of sexual dimorphism. Another is the peacock and the peahen. So this is the male over here, the peacock, and then this over here is the female, the peahen. And they look dramatically different, okay? And so in some species, individuals compete directly with members of the same sex for mates. That type of sexual selection is called intrasexual selection. Intrasexual selection. Okay, so in some species, individuals compete directly with members of the same sex for mates, which is called intrasexual selection. Okay, that would be like where most often the males are competing with one another. Sometimes physical combat, more often ritualistic displays. However, the more common type of sexual selection, okay, and that would be like where two rams are butting heads, or the deer are fighting one another. But the more common type of sexual selection is called intersexual selection. Intersexual selection, which is between sexes, or mate choice. Okay, so this is also called mate choice. What happens is usually females are more choosy. One sex, usually the females are more choosy in selecting their their mates. So what's the advantage of this? Well, one hypothesis is that it's called the good genes hypothesis, where females pick a mate based on these secondary sexual characteristics because, and a peacock is a perfect example, because the peacock is this slow bird. It doesn't fly very well. It flies, but not very well. It really kind of jumps. And they live out on the prairie in China. And so there's all kinds of foxes and stuff like that could come around and eat them. And so this guy right here with this big tail feather display carries those feathers around with him all over the place. And so he must be really, really good at surviving because he has to carry all this extra weight and all this extra stuff in order to do that. He's also really bright in color, which makes him super obvious. And so that means that his genes must be fantastic. And so that's why the female will pick those males. Okay, so a little bit more on natural selection. There are some human diseases, where the drugs that we've given, antibiotics we've given, have facilitated natural selection to occur, or at least some selection to occur, so that they are no longer able to be killed by these drugs. During the 1950s or so, there's some medical experiments on bacteria and things like that were super successful, and we thought that soon enough infectious diseases would be beat. But it didn't come true. It's because we didn't take into account evolution. So antibiotics will select for resistant bacteria. In a population, there might be one or two individuals that can beat some drug. And so you apply that drug, you kill all the individuals in the population that can't beat it, except for the one or two that can. And then those individuals start breeding like crazy. And then the ones that are left over are antibiotic resistant phenotypes. So we have contributed to this problem in a couple of ways. Number one, doctors may have overprescribed antibiotics. You go to the doctor and you don't feel so well, and so you get like a Z-Pak or something like that. And but it turns out that you actually have a viral infection, and so you've introduced a bunch of potential selection to occur inside your body by exposing these organisms to the antibiotic, and that's not good. So doctors have to be a little bit more careful in prescribing antibiotics. And then number two, you may misuse, as a patient, the prescribed antibiotics by prematurely stopping the medication because you already feel better. So the compliance of patients, that's where a patient doesn't use the antibiotic to their full amount because they already start feeling better, and so then they don't comply with the prescribed dosage. And then lastly, and this one's kind of a not on the doctor so much, okay, but the last one is that livestock, like cows and sheep and pigs, livestock, the producers, the people who grow them, they add antibiotics to their feed as something to help them kind of grow faster and prevent illness as they grow. And so they're just giving them huge amounts of antibiotics and exposing all those bacteria to antibiotics. So then we potentially could get a lot of antibiotic resistance. So these three things, these are the reason that we have these superbugs out there in this world that we can't treat. The tendency for natural selection to reduce variation in populations is countered by the mechanisms that maintain variation. So what prevents natural selection from eliminating all the variation as it selects against unfavorable genotypes? And then why aren't less adaptive alleles eliminated as the best alleles pass to the next generation? Well, in some cases, genetic variation is preserved rather than being reduced by natural selection. So sometimes what's best for the population is for genetic variation to be preserved. And so one of those is called balancing selection, which occurs when natural selection maintains a stable frequency between two or more phenotypic forms in a population. So this is where you maintain very specific sets of genotypes in the population. And the best example of this, and almost the only example of this, well, not the only, one of the best examples of this is this fish, and I'll describe this fish in a minute, but this fish that eats scales off the sides of other fish, and it's really super cool. Another a type of balancing selection, one type of balancing selection is called heterozygote advantage. This is where heterozygotes have the best reproductive success. That means that you're going to maintain heterozygotes, okay, the ones with two different alleles. And then that means that sometimes you have homozygous recessive and sometimes you have homozygous dominant individuals in the population, where the best ones, though, are the ones with the heterozygote in the system. And the best example of a heterozygote advantage is in humans, actually. This is sickle cell anemia. In sickle cell anemia, if you're homozygous recessive, okay, so you have two sickle cell alleles, then you can't properly get oxygen to your muscles in your body, and you can die. If you don't have either, okay, if you have two non-sickle cell alleles, then you have a higher propensity to die if you get malaria. Okay, so then malaria is problematic. And so in areas where malaria is a thing, the heterozygotes have the advantage. They have some sickle cells where the parasites can't get in and cause the harm that malaria causes. And they have enough regular blood cells that they can get blood or oxygen to all their tissues. And so the heterozygote does the best. But then you also get both other types of phenotypes in the environment that occur. The one that I was talking about before is called frequency-dependent selection with the fish. Frequency-dependent selection is a type of balancing selection that maintains two different phenotypic forms in a population. So there is the scale-eating fish in a lake in Africa, which attacks other fish from behind. It kind of darts in, removes a few scales from the side of its prey, and then escapes away. Okay, this fish, the prey here doesn't like that, and so it swats them away really quickly, but not before they can grab a few. And there are some fish with a mouth that kind of faces off to the left, and then there's some fish with a mouth that faces off to the right. Okay, and the frequency of those in the population is maintained, because if they were all right-sided fish, then there wouldn't be enough scales over here for them to eat, and they would do not so well. Then that would allow more of the left mouth fishes to do better. Then they eat scales, and there you go. And so then there's a certain number of each one of these is maintained in the population because of the environment, called frequency-dependent selection. It's a type of balancing selection. Okay, the evolution of organisms is constrained. Selection can only act on existing variations. So we're not just producing new advantageous alleles because we need them in the environment. If they're there, then selection can act on them. Number 2, evolution is limited by historical constraints. Evolution takes existing structures and adapts them to new situations. It doesn't produce something perfect for the environment, and there you go. It's not like because we have water bottles or something like that, the best way to drink out of that water bottle would be to have a perfectly circular mouth. It's not like that's what humans do. It's not like that's how we evolve. It's what's already there, historically, is just kind of co-opted and changed a little bit so that it can fit the new situations. Number 3, adaptations are often compromises. So the same structure often performs a lot of different functions. So, for example, my little goofy water bottle example here, it's not that your mouth is only drinking water, it's that your mouth is also chewing food, and it's also breathing air, and it's also talking, and it's used for a lot of things, and so it is a compromise between all of its functions that leads to the shape of the human mouth. The other thing is that it's not just natural selection, it's not just random chance, and it's not just the environment. It's those things interacting that lead to change. And so sometimes the environment will change unpredictably. And when that happens, then natural selection varies or heads something off in the other direction. And sometimes random chance leads to change. And sometimes, and so the point here is that sometimes you go down an evolutionary path because of something that happened in the past that no longer applies in the future. And so just remember that the environment's always changing. And so the whole point here of all of this, the main point, is that natural selection cannot fashion perfect organisms. All right, a point that's so important that I want you guys to write that down, big bold letters, underline it, and put little stars by it. Okay, so I should see that in your notes. And how about if I see it, I'll give you some extra credit points. 1 extra credit points for this homework assignment. Okay, and so with that, I'm going to go ahead and say goodbye. So With that, I'm going to go ahead and say goodbye. I hope you guys enjoyed the video. I know it was a little long, but I'll see you in class. Bye.